All right, we're at Module 25 to answer that all-important question, is God in our genes? And if so, what do we know about the genetics of religious experience? Today's module is going to be delivered to you in 3 chunks. First, we're going to talk about a guy named Dean Hamer and his so-called God gene hypothesis. Then we're going to look at some of the details, including the idea of self-transcendence and this thing called VMAT2. And then finally, we're going to do a little critique of the God gene hypothesis and where we are now with respect to the idea that there is a genetic component to belief in God. So, we all are curious about the idea that religion may have a genetic basis. We wonder if there is a set of genes, perhaps, that makes us religious. And in essence, that's sort of what the God gene hypothesis is getting at. It was originally postulated by a scientist whose name is Dean Hamer. He is not a scientific slouch. He's the real deal, a serious scientist. He is the director of the Gene Structure and Regulation Unit of the National Cancer Institute. And as part of his work, he undertook sort of a side study that wasn't part of his regular work, and from it he came up with the so-called God gene. This is the title of his book, How Faith is Hardwired into Our Genes, published in 2005. And if you sum it all up, the hypothesis that he espoused and promoted is a combination of a whole bunch of things jumbled together, including behavioral genetics, a little bit of neurobiology, and a fair amount of psychology. So if we decide that there is a genetic basis to belief in God, there are all sorts of other ancillary questions that immediately arise. Things like, did humans create religions from cues sent directly from above? Or, as is probably more likely, did evolution instill in us a sense of divine awareness, a desire to believe in a supernatural God, and did that belief somehow help to keep the human species going? We've talked several times about the reality that there were many times during our hunter-gatherer phase when the entire human population could have gone extinct. And so any advantage conveyed that kept people going, despite the many problems they faced, would have been a huge advantage. Another issue that arises if we decide that there's a genetic basis to religious belief is whether or not some people are inherently more spiritual than others. And if they are, is it genetic or is it nature? Is it nurture? We don't know. But those questions certainly arise, and they're important because this clearly is one of the fundamental aspects of being human. And so it really does go to the core of what it means to be human. Now let's take a closer look at the major proponent of this idea. In other words, Dr. Dean Hamer. He is a genuine real-life molecular biologist, and as I mentioned, he's the chief of the Gene Structure Unit at NCI. And he claims that spirituality, however you want to define it, is an adaptive trait. And to that end, he claims to have located what he's now referring to as one of the genes responsible for human belief in religion, but as you may have discerned from the title of his book, what he's really claiming in the book and the related materials is that there's one gene for belief in God. So this then gets us to the question of, okay, if there is a gene, what gene would that be precisely? And this is where the neurobiology comes into play. The gene in question happens to code for the production of some of those neurotransmitters that have a role in the regulation of mood. And in the study that he undertook, from which the God gene eventually emerged, he actually artificially confined his search to these specific genes. So that, of course, raises the question, is religious experience just another experience that revolves around particular neurohormones and their influence in the body? Pretty much the same sort of arrangement that we talked about when we discussed Helen Fisher's work. Is that what we're talking about here? And Hamer isn't saying that specifically, but he does believe that every experience human beings have is somehow or other anchored in neurochemistry. And so in this sense, the fact that religion might also share this underpinning is not a particularly remarkable one. In other words, religion is just yet another human characteristic that has its origin in the firing of neurons and the relevant neurohormones and neurotransmitters. So his quest to understand this began pretty much by accident back in 1998. At the time, part of his job was to do work on a survey about smoking and addiction for NCI. Obviously, they're interested in the relationship between smoking and its ability to get you addicted. They wanted to know, in part, whether the tendency to become addicted was genetic. And so there was reason for Hamer to be looking at people's genes when they came into his lab. As a specific part of this study, 1,000 men and women were obtained, and they agreed to take a standardized personality test, or TCI, which was a measurement of their temperament and characteristics, or character indices. And another thing that was measured during this TCI test was a trait that Hamer called self-transcendence, the ability to leave one's present reality and go somewhere else. In Hamer's definition, self-transcendence could be parsed into three traits that make it up. The first of these is self-forgetfulness, or the ability to get lost entirely in an experience. So it might be, I don't know, playing sports. You might have a love for music and just become consumed by the music that's meaningful to you. At any rate, you become, you go to a different place and you are not entirely aware of what's going on around you. You're simply absorbed into this thing that you love. The second component of self-transcendence, according to Hamer, is transpersonal identification, which he thinks is a feeling of connectedness to a larger universe. It's the sort of thing the Buddhist monks were also feeling when they did their meditation and felt that they had left their body and went somewhere else. And lastly, the third component of self-transcendence is mysticism, or things that may or may not be literally provable, something perhaps that doesn't have an immediate physical basis underpinning it. So all told, Hamer argues that the self-transcendence traits are a proxy for measuring spirituality. So he did this survey, and as he was amassing the data and analyzing the data, he got interested in a subset of the data. And what he decided to do in the end was to use the data he had obtained from the smoking survey to conduct a second study, which was the so-called spirituality study. Using the data that he had and the people that he'd already assessed, he first ranked the participants on a self-transcendence scale from most to least. And then, because he had genetic profiles on the participants, he started looking at their genes to see if he could find a gene or a section of DNA that could explain the differences in self-transcendence that he saw in his survey. But here he had a problem. He had a lot of genetic data. He may have had the full genetic profile of each person in the study. Today, that's a really easy thing to do. Back when he was doing this, it was much more difficult. It would take quite a long time to get the entire genetic profile of a single person. But now it's less than a day's work. We know that it's a daunting task, and it was particularly back in Hamer's day, because there are roughly 35,000 genes in the human genome, which means there are roughly 3.2 billion base pairs comprising those genes. So if you're going to go base pair by base pair through the entire human genome of each person, it's going to take a long time, particularly back then when the technology wasn't as advanced as it is today. So to kind of move things along and narrow down the possibilities, and here's a critical decision in the study. Hamer decided to narrow his search to 9 specific genes, the location of which he knew in the genome, and these 9 specific genes all coded for the production of particular monoamines that can serve as neurotransmitters in the nervous system. So I ask if this is a problem. I think it is, because he looked at 9 genes out of a possible 35,000 genes, and we don't know that there might not be other more explanatory genes in the human genome other than the ones that he looked at. But he knew where these genes were, he had a way to assess them, and he thought, based on what he knew about neurobiology and neurochemistry, that these genes would logically play a role in self-transcendence, spirituality, and so forth. So the genes that he included in his analysis included things like the ones you know, serotonin, norepinephrine, and dopamine. So the basic idea was to focus on human genes responsible for the production of neurotransmitters that regulate aspects of your mood. And with that narrow subset of data, he quickly noticed that there was a variation in a specific gene. The gene was called VMAT2, which stands for vesicular monoamine transmitter. And the variant, or allele, as we would call it, an alternate form of a gene, seemed to be directly related to how volunteers scored on the self-transcendence test. Now, the particular variant or allele of that gene had a cytosine substitution in a particular place in the gene, so they could identify the allele quite easily. And the variant, the people with that variant of the gene, ranked particularly high on self-transcendence. So interestingly, a single DNA base change in the middle of a specific gene had a direct correlation to levels of transcendence in his test population. So with that, he put together the God gene hypothesis, and these are the basic tenets of that idea. 1, spirituality can be quantified by psychometric measurements, things like the levels of the neurotransmitters that we're talking about. He also argued that the tendency to be religious is heritable. In other words, it's genetically coded, passed from parent to offspring. And perhaps the most important thing that he was adding to our understanding of this whole process is that the VMAT2 was part of that heredity and part of what makes humans susceptible to or desirous of having a relationship with a supernatural God. He argues further that VMAT2 acts by altering monoamine levels, and that increased monoamine levels lead to spirituality, which was favored by natural selection back in the day, and therefore people who had it did better, left more offspring, and so forth. A little bit more on the details of VMAT2. We've already talked about the name, but what does it do exactly inside the body? Well, it produces a protein that is lodged in the cellular membrane that surrounds each cell, and its function is to transport monoamines, such as dopamine and the others that we've studied, from the cytosol, that's to say the solution that exists inside the cell that bathes all of these cellular components. And what happens with VMAT2 is it helps to transport monoamines through the cytosol and eventually enclose them in synaptic vesicles. You recall from our discussions back in, I think it was Module 5, when we talked about neurotransmitters and how they worked, what we said was that once an action potential gets to the end of an axon, it causes for vesicles filled with neurotransmitters to fuse with the axonal membrane, thereby dumping their neurotransmitters into the synaptic cleft. Well, obviously, if that's going to happen, you have to get the neurotransmitters packed into those synaptic vesicles, and that's what the VMAT2 is doing. It's helping to package the monoamines in these vesicles so that they can later be used for neurotransmitter purposes. So in humans, we know that the gene that encodes this protein has a particular name, and there it is for you if you're curious about it. So again, the neurons work by using neurotransmitter substances at the terminus of a particular thing going here, at the terminal end of an axon. Once the action potential reaches this end, it's going to cause one of these vesicles to fuse with the axonal membrane. Here you see the contents of the vesicle spilling out into the synaptic cleft. The little neurotransmitter molecules are going to bind to this dendritic protein receptor on the other side of the synapse. Stop that. And that is how the message is passed from neuron to neuron. So it's really important. You wouldn't be able to function at all without this happening. So if we use that general model to describe what VMAT2 does particularly, we know that VMAT2 is essential in the presynaptic neuron and its ability to package and then subsequently release the neurotransmitters into the cleft. If the VMAT2 gene is inhibited or damaged, the neurotransmitters, and this can be more than one kind of neurotransmitter. It could be dopamine, could be serotonin, could be epinephrine, could be norepinephrine. There are lots of different neurotransmitters that rely upon this process. So if it's inhibited, not enough neurotransmitter will be released, and that causes problems everywhere. Impaired VMAT2 might be a physiological basis for depression or anxiety or many other conditions that relate to low levels of neurotransmitters. Some of these include really bad diseases like Parkinson's and so forth. We also know that chronic cocaine use shows a significant reduction in VMAT2, which is yet another reason not to use cocaine. A fair number of animal studies have been done on VMAT2. Among the more interesting ones, we have the findings that mice bred without VMAT2 experienced intense depression, although I'm not sure how we measure that in mice. Probably it's simply hypoactivity, low levels of activity when you expect regular, normal activity. And finally, they die rather quickly because they simply can't function without their neurotransmitters. Also, the brains of these mice show significant depression in a series of monoamines. So it's not just dopamine, it's not just one, it's many. And additional studies have shown that the monoamines were present in the cells and the axons of these animals, but they were not packaged in vesicles and therefore were not functional. Some more specifics pertaining to Hamer's study. He says that VMAT2 does not encode for belief in God. Rather, he argues that VMAT2 encodes for a physiological state, presumably neurotransmitter-induced, that produces sensations associated by some people as the presence of God. He says further that this tendency towards spirituality can be adequately quantified and captured by the self-transcendent scale, and as a result can be measured by psychologists. He goes on to say that neurotransmitters, as a result, in all likelihood play a major role in regulating the brain activities associated with mystic beliefs. So his general hypothesis here is in red, namely self-transcendent people are more optimistic due to more or better release of monoamines, and as a consequence lead healthier lives, leave more offspring, and have a greater capacity to do this because they are self-transcendent and many are religious. When pressed for a conclusion, Hamer said that the genetic variant of VMAT2, the one with the cytosine substitution in the middle of the gene, were more likely to be self-transcendent, and that, in fact, was borne out by his survey data. To the extent that self-transcendence is required to be spiritual, these individuals, those with the variant of VMAT2, would be expected to have a greater capacity for spirituality. Now, he was less interested in taking the next step and arguing that as a result of having this increased capacity for spirituality, they also believed in a supernatural God. But then he published the book, and I'm pretty sure that the publisher was responsible for the title, which, as you may recall, read The God Gene, the singular God Gene. Now, the publisher wanted to sell books, but Hamer, as a pretty good scientist, knew, I am sure, that that was pushing the data to a level that was not supported. And as a result, he met with wild, rampant criticism, and furthermore, it came from both sides, from those who believe in God and from those who did not believe in God. So there was a media circus. He did all the rounds on the talk shows with Oprah and all those people, Terry Gross. They all wanted to know about the God gene, but as he talked and as people reacted, it was pretty clear that he had some problems, and that as a scientist, he was going to be unable to maintain the God gene hypothesis, because the first thing he had to admit publicly is that human behavior, particularly complicated human behavior, like belief in God, is determined, if it is genetic at all, it's determined by many genes acting in concert. So if we were, it's as if you wanted to find an IQ gene. You could certainly find not one, but many genes that have some role in producing what we eventually call IQ. And religious spirituality is a complicated state. So it too is, to the extent that it is genetic, also the product of many, perhaps as many as hundreds or thousands of genes. There just is no single God gene. So what Hamer may have done in his study is to have located one genetic component of spirituality, but that is not the same as saying he identified the gene that is responsible for religious behavior. Furthermore, Hamer would make a distinction between spirituality, which is personal, it's not prescribed particularly, and religion, which is institutional. There are sects, that's S-E-C-T-S. There are specific doctrines that particular religions have, and as a result, you can't talk about religion as a single entity either. There are many different religions, and within religions there are many different denominations and so forth. So religion is institutional. It depends upon the supporting institutions to have its characteristics, its flavor. and so forth. So from a scientific standpoint, it's easier and probably more accurate to talk about spirituality rather than religion. Well, now in chunk 2, let's take up a little bit more investigation of this idea of self-transcendence in humans. There have been a limited number of twin studies, which you probably know is the typical way that we try to look at human behavioral traits whenever possible. If you can find identical twins, meaning that they were the product of a single egg, fertilized egg, that split early in gestation into 2 eggs and then proceeded to become 2 children that both had identical genetic constitutions, if you can find those kinds of kids who are then separated at birth and raised in different environments, this gives you a way of assessing that which is genetic and comparing it to that which is environmental. So in such studies, there has been shown to be some heritability to religion. In other words, it was much more likely that twins separated at birth would share a similar religious inclination than for twins who did not have genetic constitutions that were the same. However, the same studies showed that specific religious beliefs, that is to say the institutional part of spirituality, had no genetic basis and was culturally instilled. So that's still consistent with what Hamer is saying. But we also have to go back to precisely how we arrived at VMAT2 as the identified God gene, because as I alluded to earlier, there are some problems with the way he conducted the study. It's understandable that he did it this way because he hadn't begun the study with the intention of resolving the question of spirituality. He had a mandate to look for a link between smoking and addiction. This kind of came out of that project as a side project, but one that was not called for by the sponsor of the research. So for that reason, he was very limited in what he could do. He had to get by with the data he had collected for another purpose, and that meant then that his conclusion was going to lack a sweeping basis when he got done. At any rate, if we look rigorously at what he did, he analyzed DNA and personality scores for 1,000 individuals. And as a result of that study, he found a significant correlation with VMAT2 and spirituality, defined as self-transcendence. So here's the scientific criticism of what he did. We know that VMAT2 does appear to be involved in monoamine transmission. We've talked about the mechanism. Monoamine transmission may indeed be involved in religious experience as well as spirituality and mystical feelings generally. But monoamines are also transported for dozens, if not hundreds of things, without which you would die, like the mice that we talked about earlier. So the fact that there's a correlation of some sort between a particular gene and its products with spirituality is fine, but it doesn't necessarily mean causation. It could be simply something that's correlated, but it lacks a causal connection. We just don't know. And of course, the monoamines are in the cells specifically to do other things, and it may be the case that the monoamines have this ancillary effect that results in enhanced spirituality, that may or may not have been part of the original repertoire of things that the monoamines were designed to do. At any rate, it is categorically wrong to consider VMAT2 to be the God gene, A, because, as I mentioned before, there are many genes involved in spirituality, and B, because VMAT2 does a whole bunch of other things other than whatever is going on with spirituality. One scientist put it in a fairly succinct way. What Hamer found with respect to VMAT2 is, quote, a gene that accounts for less than 1% of the variance found in scores on psychological questionnaires designed to measure a factor called self-transcendence, which can signify anything from belonging to the Green Party to believing in ESP, according to one published, unreplicated study. So that pretty much summarizes where we are here. We have a gene, and whatever it's doing in this study that Hamer did, it's only controlling 1% of the variance in the scores. So 99% of the variance is due to other stuff. And that's not the most satisfying result for a scientific study. And of course, the fact that this is unreplicated, the hallmark of science is that you have to be able to replicate it. No one has, so here we are. Some folks from the religious side of the ledger weighed in on Hamer's work as well. One of them, John Polkinghorne, is a very well-known Anglican priest, very highly respected, not only by those of faith, but by those who are not. He said, quote, the idea of a God gene goes against all my personal theological convictions. You can't cut faith down to the lowest common denominator of genetic survival. So Polkinghorne just reviles at the idea that Darwinian selection had anything to do with religious experience. Another person, Walter Houston, who's an Oxford chaplain, said, quote, religious belief is not just related to a person's constitution, it's related to society, tradition, character, everything's involved. Having a gene that could do all that seems pretty unlikely to me. And I think most of us agree with that. So in the end, Hamer was getting beaten up pretty regularly by both the scientists and the men of the cloth who thought that the God gene idea was just too simplistic and naive to hold any water. Certainly the notion of a single gene is a problem, and since publishing the book and going on all the talk shows, Hamer has seriously backtracked on this issue and admits that many genes are no doubt involved in religious expression. He agrees with the clerics that culture is involved, and at some level natural selection may be involved in the production of the trait and the expression of the trait in a way that ends up having a relationship to spirituality. It's also likely that group selection is involved, but that is an argument for another day. So we're still left with the question, is God in the genome, in your genes? We don't know yet, but we'll keep asking, we'll keep looking. And here's your study guide for Module 25.